Good morning, everybody. Happy Monday. Get stuff going. All right. So, before we move on to DNA structure and replication, pardon me, does anybody have any questions about linkage? Especially working out 3-point crosses. Now would be a good time to ask. Cat fur everywhere. All right, well, if anything does come to mind, give me a shout and I'll go through it. So basically what I want to do today, let's see where we're at in the schedule. It's that one, I do believe. That's Monday the 4th already, crikey. So we're a little behind. We might actually end up changing the exam date to this Friday, but next Friday. So you can see it's scheduled for Wednesday, but I think we're about a day behind from last section. So we'll see how it goes, but that'll probably end up changing. So yeah, basically what we have now is to get through DNA structure, modes of DNA replication, and that's in both prokaryotes and eukaryotes, and also even viruses a little bit. And then get into the mechanics of homologous recombination. So first up really is a kind of a recap, to be honest, of a lot of stuff, because it's really important to get kind of several key points in DNA structure to be able to understand how it's copied. And really one of the biggest deals, and this is a replication fork in eukaryotes. Yeah, and I can tell that because of the different names of the DNA polymerases. One of the key things to get is directionality. Right? DNA has a direction, as does mRNA, as does protein. And that direction is super important for replication and for transcription and translation. Right? So these molecules have essentially a beginning and an end. And DNA is especially weird because it has two beginnings and ends, right? And they're going in opposite directions. And so we need to kind of get that clear because it really impacts a lot of other stuff that happens. So actually, I can't see there. Yeah, move that over there. And so some of this may seem a little repetitive, maybe stuff you know already, which case that's awesome. It may be stuff that actually helps you understand this process better, which would be great. So essentially what we're talking about in terms of DNA are a series of repeating molecules called nucleotides, right? And a nucleotide essentially consists of 3 parts, right? A phosphate group, which is charged, hence DNA having a charge, right? And it's a negative charge. So that's important later on when we look at higher order structures of DNA, such as chromatin and its control. We have a 5-carbon sugar, that's the ribose in deoxyribonucleic acid and ribonucleic acid. And one thing to really kind of emphasize, and I'll just zoom up a little bit here, if we do, is that these carbons are numbered. They don't actually have numbers stuck to them, obviously, because they're atoms, but they are numbered clockwise going round from the oxygen. So I always think these look like a cupcake, actually, with a cherry on top, where the cherry is the oxygen. And you always count the numbers from the carbon to the right of the oxygen clockwise, as if you're counting the time. And so these 5 carbons, kind of inherently in and of themselves, they're not super important, but which carbon has what attached to it is very important. So in on the first carbon is attached the nitrogenous base, right? That's the part that's different between nucleotides, between adenine, cytosine, guanine, and thymine. In DNA, the third carbon has a hydroxyl group, right? And I'll use these terms a lot. Hydroxyl is essentially a OH group. And this is essential for extension of the DNA and RNA chain, right? Because that is where the dehydration condensation reaction occurs. It's basically required for the formation of a phosphodiester bond. Bond. James Bond. My name's Bond. James Bond. Actually, that's more of a Mancunian James Bond, of which there are none. Actually, I'm very much looking forward to seeing the latest and last James Bond. I feel a little sad that Daniel Craig is hanging up his hat, but hopefully the next James Bond will be equally as cool and rugged as Daniel Craig. Bit of a man crush, a little bit. I mean, who wouldn't, right? He's a bit of a hunk. So anyway, moving swiftly on. This 3' OH is really, really, really important. And it's one of the key parts of how we denote direction in DNA and RNA. All right. Then the next one is the 5' carbon. So 1' carbon equals nitrogenous base. 3' carbon equals OH group. 5' carbon equals phosphate group. Right, so those are the 3 main ones. Now in RNA, the 2' carbon also has a hydroxyl group. And that's kind of important for other things, in particular splicing in eukaryotes, which we'll get to in the which section is that? 3rd section, I think. Okay, so that's the basic structure, right? So that's a nucleotide, consists of those 3 things. These 2 things, the phosphate and the sugar, are the same amongst all nucleotides. They only differ really between RNA and DNA, right? The bits that do differ are these nitrogenous bases. And I love this picture, or this graphic, whatever you want to call it, partly because it helps explain, well, for one, it shows why they're called nitrogenous bases, right? Because they've got a whole bunch of nitrogens in them. But it also shows you why base pairing occurs, right? So everybody remember what adenine base pairs with. A little bit of a recap. What does adenine base pair with? Thymine, yeah, exactly, right? Guanine with cytosine. So now you can see why, right? Adenine has, what that would be one slightly positive and one slightly negative atom or region. And thymine has the matching opposite, one slightly negative, one slightly positive region, right? These are because of polar covalent bonds between oxygen and something and nitrogen and something, basically, right? So just looking at this picture, you can already tell how many bonds, hydrogen bonds, hold adenine and thymines together. How many do you reckon there are? Also another bit of a recap. There's two, right? Because there's only two of those regions that will work to form hydrogen bonds, right? So that's the molecular or even, I don't know, biochemical basis of the number of hydrogen bonds between bases. So that's, I think it's kind of neat. You can also see the same for guanine and cytosine, right? We've got 3 plus plus minus, and then 3 minus minus plus, right? And so those match perfectly, right? Literally like kind of lock and key together to form those hydrogen bonds. This base pairing, this specificity, is a large chunk of the fidelity of DNA replication, right? And so in every single one of your cells, you have pretty much exactly the same DNA. They may change by like 1 or 2 bases, right? Out of, what would it be, 6 billion total per cell, because of the fidelity of DNA replication, right? You don't want to have a body with cells that have different DNA in them, because all kinds of weird and wacky stuff would happen, and typically none of it good, right? So a lot of what we're going to be talking about next, kind of like higher order stuff, all relies on this, right? Base pairing and structure, right? And in fact, a lot of the stuff that we talk about, just bigger picture type things like epigenetics, right? The non-DNA sequence information you inherit from your parents and grandparents, right? That depends upon the charge of DNA for it to, do what it does. So really, it all comes back to this. That's why I want to spend time on it, right? Because it's really important. Now, again, obviously, you've seen this picture before. This is another recap. Main point here is, well, a couple of points, actually. One is that this is called the phosphate sugar backbone, right? And so this is essentially if you think of DNA as a ladder, right? And it's not a perfect analogy, but it'll do, right? The phosphate sugar backbone are the uprights. The nitrogenous bases between are the rungs, right? And so in many ways, this is actually what makes DNA such a stable molecule, both in the cell and also in kind of history, right? Archaeology. They've, oh, actually, you know what's really cool? Totally unrelated. This is more population genetics. I actually read this morning while I was eating my granola, funnily enough, that some population geneticists have actually worked out the order of migration in the Pacific. So, shoot, what was that film about Maui and played by Dwayne Johnson? One that has "You're Welcome." It's okay, you're welcome. Da, 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 da. Moana, that's it. Thank you, Brianna. Just couldn't remember the name off the top of my head. But anyway, so you all know about the migration from, well, actually, it was originally from Taiwan, which is crazy. I didn't know that, to Fiji and Vanuatu, I think, is the other kind of big island. But anyway, they've actually worked out the order of migration, those absolutely crazy, like sail off into the nothing, essentially, trips of those Polynesians. They've worked out the order by looking at the genetics of existing inhabitants of those islands. And they've actually figured out the different waves of migrations. And the one that was the last was to Easter Island. And it occurred over about 500 years, all based on population genetics, founder effect, essentially. Basically, the genetic diversity decreased at every step because there's only a limited number of people in the boats. Isn't that crazy? I think that's super, super interesting. Completely like an aside, but essentially based off what we're learning now. Also, get this as well. This is also a super, super cool fact. The last migration was over, I think, 1,100 miles. Imagine doing that in an ocean with no maps and just the food that you're carrying on board, plus whatever you can fish out of the ocean. Literally just, I mean, how many of those people missed, right? Easter Island is not very big compared to the Pacific. So anyway, I thought that was super cool. Just another application essentially of population genetics. So anyway, this phosphate sugar backbone is one of the reasons why DNA hangs around so long, why it is so stable, right? Because it is very durable and it protects the information it holds within essentially those nitrogenous bases, which are, because of their hydrogen bonds, much more reactive. So that's one thing. Second is that the direction comes from, and let's going to zoom out a little bit, the arrow is important, the order of those carbons, right? So the 3' end is where there is a 3' hydroxyl group literally kind of hanging off in the breeze, right? The 3' end is the end at which new DNA molecules are added, right? So this is almost always comes up as a question in the exam, just because it's so important. But the 3' end is where new DNA, RNA nucleotides are added. You cannot add new DNA nucleotides to the 5' end, right? And so DNA, RNA is both synthesized and read in the 5' to 3' direction. Right, and that's super, super important for all kinds of stuff, right, that we'll be learning about at all kinds of different levels, right? Even up to and including the control of gene expression, which we'll be talking about much later in the course, fourth section. And finally, as well, these nucleotides are joined by phosphodiester bonds, right? And so those phosphodiester bonds are basically between the oxygen of one nucleotide and the phosphate group of the next one. And actually there's another one within there. That's why it's called a diester bond, because there's two oxygens involved. But this is what holds them together. These are the bonds that are formed when you add a new nucleotide to that DNA strand. All right, a little bit more recap. Anti-parallel, as you already know, right? So the 5' end, if that's up on one side, it goes the other direction on the other strand. So essentially, and this is another cool thing which we'll see later on, DNA contains information in both directions, right? So the base sequence, the nucleotide or the nitrogenous base sequence, is not unidirectional, right? So the A, what does this be? That would be an A, C, T, G, right? Information that applies to one direction. You can have different information essentially encoding other proteins or non-coding RNAs or stuff going in the other direction, right? So this is an anti-parallel piece of information. What else is important here? I've already pretty much looked at this. Oh, another point to point out, the number of hydrogen bonds matters, right? So which of these two do you think would be easier to pull apart physically or enzymatically or whichever, right? Which of these two, guanine and cytosine or an adenine and thymine, would be easier to pull apart? Questions to the class. Which would be weaker, GC or AT? AT, good job, Isaiah, right? Why? Isaiah, Keanu, anybody else? Yeah, it's got fewer bonds, right? It's only got 2 versus 3. So which bases are you more likely to find in areas of DNA that need to open, right, for replication or transcription? Yeah, exactly. A's and T's, right? Here's another different way of thinking about it, a fun one actually. One of the courses I teach, well, have taught, astrobiology, it's all about extremophiles, right? And extremophiles are just super neat. So some of these extremophiles live at astonishingly high temperatures, as the record is, I can't remember what the latest one is, but I know one of them, a bacterium called, or archaean maybe, called Pyrococcus furiosus, right? So that's basically angry flame balls is what that means in Latin, is able to live at temperatures up to just under 120 degrees Celsius, right? So that's the temperature at which you would be a soggy mass of stuff, right? Melted everywhere and a bunch of bones. What do you think you're going to find more of in its genome? GCs or ATs? Yeah, GCs, exactly, right? So extremophiles that live, or hyperthermophiles actually is the better way of saying it, that live at really high temperatures have a higher GC content in their genome, right? Because they literally need those stronger bonds to physically hold their DNA together at those higher temperatures, right? So it doesn't fall to pieces, essentially. And a whole bunch of other changes to their lipids and especially to their proteins too. Super fascinating topic, really is. Anyway, so this is a pretty fundamental property of DNA, and you will see as later on when we get into kind of the sequence hypothesis and gene expression, the regions that we deal with involved in transcription are typically AT rich, right? Because they have to be opened frequently. That's like the purpose of it. What else? Some numbers. Again, actually, this is what I want. It's a non-symmetrical twist, which is kind of interesting, right? So there's a minor groove, which has a smaller gap between the opposing strands, and a major groove, which is much wider. And proteins that bind DNA, they recognize typically 2 things. One is the DNA sequence, of DNA, right? So they only bind to specific spots. And that's super, super important. And two, they actually recognize the topography of DNA, like its shape, right? And so a good example is, oh man, lacking the tryptophan. There you go. I should remember this, right? The TRPR, so a repressor protein in the tryptophan operon, or the TRP operon. It actually binds two major groups. So it kind of comes in and binds separately two regions spaced exactly one major groove apart, right? So that's another important part of that. Okay, so when we get into DNA replication, right? So there's a lot going on. Again, there's some more words to learn. And here's some of them. So essentially, there are very specific places at which DNA replication begins. Bacteria have one in their circular genome. Eukaryotes with linear chromosomes have many, thousands potentially, right? So at every one of these spots will be typically an AT-rich sequence that's recognized by the transcription machinery, right? So it starts. Once that gets going, that generates a replication fork. I don't know why that's not underlined. Yeah, nice and tidy, right? Which is where essentially the double helix gets separated and the new DNA starts getting synthesized, right? And there's a whole bunch of proteins. Essentially, this up here is a picture of a replication fork, right? So the original parental DNA here, bunch of proteins, and then you get your two newly synthesized daughter strands. Essentially, your sister chromatids are coming out this other end. And when you have that form, you get what's called a replication bubble. And so this is literally, I wonder if I have a picture of it. No, I don't have a picture of it in this PowerPoint. I thought I did in the other one, maybe. Ah, that's a shame. I should dig a picture out. It's really kind of neat. It actually physically looks like a bubble, right? Which is essentially, as those replication forks move, you get this kind of expanding bubble, right? It almost looks like a blister, right? Which is either side of the bubble is the original parental DNA. The bubble is then formed by the newly synthesized DNA. Right. Okay, anybody got any questions so far? Kind of mostly recap, but hopefully with a few extra bits of info and kind of perspective that help. I'm also very happy I managed to get another animated film reference into biology. That is actually a very cool film. It's lots of fun, but it is actually somewhat, it is based on kind of history of migration of the Polynesians as well. Okay, so I also really like this picture. I love good graphics. Good graphics are really hard to find in teaching. So let's see if I can blow this up. This image is awesome, in my opinion. Let's see if I can. Okay, so contains a lot of information. It's also very accurate as well, right? So we even have the minor and the major groove here, right? This larger open twist being the major groove. We have a anti-parallel structure, right? So we have a 5' strand, which if you follow my cursor kind of goes all the way down there. So 5' to 3'. And then you have the other anti-parallel strand starting down here, 5' going all the way up to up here where the 3' end is. So that's neat. We have a replication fork, right? So where the DNA, the parental strands are separated, right? And so here's another bit of terminology. Parental strands are essentially the original DNA that needs to be replicated. And the daughter strands are the newly synthesized DNA. So that's neat. Also shows you that the synthesis is occurring in a 5' to 3' direction, right? So the, actually, here you go. See if you can see this as well as I can. What's the next nucleotide that's going to get added to this daughter strand, the one on the left? It's going to be a G, it's going to be a guanine, right? You can't see the one that's going to be added to this one because obviously it's off the screen, right? So what you're getting here is the exact copy of, well, not the exact opposite. There you go. That's probably a better way of putting it, of the parental strand being copied. Now that is, so this daughter strand here, if you look at the sequence, is an exact copy of the other parental strand. So by the time this is all finished, right, it's kind of the zipper kind of goes up and everything gets synthesized, you'll have 2 new DNA molecules. Each one will consist of a parental strand and a daughter strand. So of course, sister chromatids, really that's a eukaryotic thing. So I just have to be a little careful to point out that this is the same in bacteria as well, right? So it consists of 1 parental strand and 1 newly synthesized daughter strand. And the other one is essentially the opposite. So the other thing that this picture shows really well is that DNA synthesis does not occur in the same direction for both strands. And this is a key point, especially in eukaryotes, right, with their linear chromosomes. Prokaryotes is a little, it's a little different depending on how their DNA is replicated. And so this daughter strand is going up because it's being synthesized 5' to 3'. You end up with the same anti-parallel structure, right, as the original parental strand. The other one, so this would be if the replication fork is going up, which it is in this case. This daughter strand would be the leading strand. So if the replication fork's moving up the page, the strand that gets synthesized up the page essentially is the leading strand. The one that gets synthesized in the opposite direction that's called the lagging strand. right? Be really, really clear. It's the lagging strand, not because DNA synthesis takes less time or longer, right? It's the lagging strand because more has to be done to finish or process the DNA so that it is fully complete, essentially. The actual adding of new nucleotides takes the same speed for both, right? Okay, so you'll come across this kind of language again when we look at transcription and translation, right? Essentially, it's the idea that these processes have phases, right? Essentially, it's like a book. You know, you have a beginning, a middle, and the end, right? And in the case of DNA replication, you have the initiation, right? You've got to start it, and it has to be started at specific spots, those being replication origins. You have to unwind the DNA, right? So you have to physically separate it. You elongate it, and then you end it, right? So there are many, many things that are required for DNA replication. And if you think about it, this is really why the GAP1 phase of the cell division takes so long, because you have to make all of this stuff, right? You have to make all of the dNTPs, right? You have to, in many cases, physically, biochemically synthesize them. So you can't make DNA without all the A, T, C's, and G's that you need to physically make the new strand, right? It takes a lot of energy to make that. You also need a whole bunch of proteins. And that's just some of them here, right? We'll get into what these proteins do and, at what point they act in a little bit. So really, GAP1 phase is all about making all of this stuff, right? And it's a lot of energy to make all of this, all of these proteins, nucleotides, the ATP required for the actual mechanism, the process itself, and so on and so forth. And obviously, you also need a single-stranded DNA template, right? Because you cannot make a triple strand of DNA. You have to separate those two strands so you have two single-stranded DNA templates. So although all DNA replication is semi-conservative, right? You'll always get one parental and one daughter strand in every, anti-parallel double helix. But how that's done varies, right? And it depends based on what those chromosomes look like, right? And so all bacteria, all plasmids, sorry, plasmids are maybe a word you've come across before, may not be. Plasmids are small, circular, essentially chromosomes in bacteria. They're also quite typically the things that get transferred from one bacterium to another that spread things like antibiotic resistance, right? Or pathogenicity traits, like the ones that turn E. coli from a fairly benign gut-dwelling bacterium into a, nasty ass bacteria that could potentially kill you, right? Those traits are typically carried on these small circular chromosomes called plasmids. Also, the main chromosome in bacteria and archaea are circular. Some viruses have circular chromosomes, others have linear chromosomes. Viruses are just really funky things anyway, just in general. But eukaryotes, that's like us, essentially have linear chromosomes that has a pretty major deal when it comes to replicating those chromosomes, because you have different challenges involved. It's also, in eukaryotes at least, one of the reasons why we get old, right, is the end replication problem in of our linear chromosomes, which is actually pretty interesting. Now, for circular DNA, we've got two different modes of replication, right? We've got what's called theta replication because of the shape that it makes, and something called rolling circle replication, right? Essentially, theta replication is the main chromosome of bacteria. Rolling circle is more about viruses and plasmids, and you'll kind of see kind of why in a little bit. And so this actually looks like the theta sign. I don't know if I can, where's the symbol? Yeah, it doesn't let me insert any. Ah, well. Where's that? There we go. That's the Greek letter theta. Ta-da! Anyway, that's why it's called theta replication, because it looks kind of like that. And essentially what happens is you have your original circular double-stranded DNA chromosome. Bacteria only have one replication origin or origin of replication, right? And then that DNA unzips, whether it's a single unidirectional or bidirectional replication, you know, it kind of doesn't really change the look, it just kind of changes how quickly it happens, basically. And then that replication bubble essentially expands and expands until you end up with two new circular chromosomes, right? That's why it's called theta replication. Now, rolling circle replication is all about speed, right? It's really all about speed, which is why, if you think about it, it's really ideally suited to viruses, right? Viruses, this is DNA viruses, right? Not RNA viruses like COVID or flu or HIV, right? Those are different and weird, essentially. Lots of biology is weird. There's actually not very much normal biology, in my opinion. But for DNA viruses, the aim of the game is to generate as many new genomes as possible, right? Because essentially being a virus is all about using your host cell to make as many more of you as you can before you get detected and killed, right? That's the kind of main drive of a virus. It's kind of what it thinks about if it had thoughts. And so rolling circle replication is super, super, super fast. Essentially, you only synthesize one strand at a time. And you do so by cutting or nicking one of those strands, not both of them, right? It's not a double-stranded break, it's a single-stranded break. Pardon me. And then you essentially replicate that, and it forces off that broken chromosome or broken strand, really, right? So you get that linear little bit of DNA, right? Because remember, it was cut back here. That then gets recircularized, right? So it kind of goes back as a circle, and then it's single-stranded, so it then gets synthesized into a new chromosome, right? While that's going on, you can do the same thing over and over and over and over again, right? So essentially, you can do multiple rounds of synthesis all at the same time, right? Which is super efficient in terms of speed. Not so great in terms of replication fidelity, but viruses aren't as concerned about that, typically. This is also something that's tied into the mechanism of genetic transfer in bacteria too. Right, so they actually feed, I think, I'm not much of a bacteriologist, but the plasmid that's being transferred, this F factor, actually gets fed into a little tube joining the bacteria together, right? And so it kind of pops out the other end, circularizes, and then gets copied as a result. So this rolling circle replication is also part of how bacteria transfer info between themselves. Which is why, one reason why bacterial antibiotic resistance is so scary is not just that the bacteria that have that resistance can evade killing by that antibiotic, but they can also transfer that trait or that ability to other bacteria too, right? So you can get that horizontal transfer. So bacterial antibiotic resistance can actually spread really fast as a result. Okay, any questions so far? Kind of nearing the end of the class. Yep. Okay, now DNA replication in eukaryotes is very different. Same principles apply, right? Semi-conservative, replication origins, so on and so forth, right? The mechanism is also in many ways largely the same too, right? Even though the enzyme names are different and there are a few wrinkles. But the big difference is that chromosomes in eukaryotes are linear. So one, DNA is always replicated bidirectionally in eukaryotes, right? You don't never have this unidirectional replication as you do in some bacteria. Also, eukaryote chromosomes are huge, absolutely enormous. So for example, some bacteria, kind of depends on what kind of bacteria you're looking at, whether they're, you know, kind of living in an environment or if they're pathogenic or whatever. But they range from 20 million to 100 million base pairs, give or take. I think that's about right, give or take. Right. Now, that's in terms of the whole genetic content. Now, we as individuals, every one of our cells, because we're also diploid, not haploid like bacteria, we have about 6 billion base pairs. Spread amongst all of our chromosomes. Now, if you think about that, that means that each chromosome is going to be millions of base pairs long itself, right? And so there's no way that you can synthesize a eukaryotic chromosome by starting at the middle or the end or whatever and just kind of going along until it's done. And so each eukaryotic chromosome has potentially thousands of replication origins, at which point DNA replication begins, right? And so essentially what you have is thousands of replication bubbles. It's like blisters essentially, right? And they will just expand until they run into or merge into the one next to it and so on, until eventually all of those replication bubbles merge and you get your two sister chromatids. All right, I'm going to leave it there. We've still got a little bit of stuff to go through, but we can do that next class. And that's it from me. So let's just move me out of the way. Make sure you get all of that stuff, right? It's very much foundational information. And we'll be getting further into the weeds on Wednesday. All right. Have a lovely day, everybody. Enjoy the cooler weather and see you either tomorrow or on Wednesday.